1 second. All right, so I'm going to start pretty much in the same crucial point where Luisa was a few minutes ago, but this time bring a computational perspective on things and just say that we look at bacteria as a wonderful source that brings computation, communication, and control together. So when we talk about this biofilm that was shown here and was observed by biologists, what we see things that compute, that communicate, and that interact in order to make this possible. So what's the greatest thing about this is that at the end of the day, we can potentially see a network that connects all these bacteria together. In other words, we can change the view from a node-centric to a network-centric perspective. And what's more important, we can see some hints toward the potential social life of these communities of bacteria. So we can talk about maybe socio-microbiology as a future session in this conference. So my talk will be organized in 3 parts. The first one is the obvious one that looks at the nodes of this network. So essentially, I'm going to talk about the computation, modeling of the computation that happens in bacteria. And this will be the intracellular part. This is probably the vast majority of the research that is presented here in the conference, and then move toward the second part, which is the intercellular networking. So in other words, we'll not look anymore inside the cell, how the signaling path goes, but how those paths get involved with the environment in order to change the behavior of the bacteria and their communication in this intercellular modeling. Finally, in the third part, since we build these networks at nanoscale, what can we do with them? Can we control them? Can we use them to our advantage to enforce a certain type of behavior and repress other type of behavior? And from here, I'm going to go a little bit more also into some thoughts for creating the hardware platforms of the system. So in other words, will be a view when the biology will inspire new computational platforms in the multi-core settings. So the first part is obviously relying on Luisa's presentation, which talked about the quorum sensing as a fundamental mechanism for communication in pathogens. And what's going on here is we use these signaling molecules that Luisa defined nicely for gram-negative and gram-positive bacteria. So in our case, as you see in this representation, there are these small molecules that are emitted by each bacteria. So this is one of the cells. And as long as this concentration is low, doesn't go beyond a certain threshold, these molecules don't have much of a role to play. However, on a certain threshold, so as I said, bacteria tend to determine that threshold and the density around them, density of those molecules around them, a coordinated response triggers. And this is where a lot of interesting things to us happen. So in this representation, these molecules are inside the bacteria. They are around 10, 20 nanometers, so they can go freely through the pores inside and outside the bacteria. So they are really at nanoscale. And essentially what they do, they connect this loci with this LASR. So this is a synthase, essentially an enzyme, and this is a receptor where this molecule gets fixed and create this complex that is represented with C in this representation, LASR AI, that is similar to putting the right key in the lock and connecting this path between these two genes that lead to all these virulence factors that are the ones that get people sick. So essentially, when this happens, this path allows the expression of both genes here, and all these factors that are in expression of the virulence become active. So what's the problem in this case? The problem is that for each of these molecules and for each of these receptors, we can write ordinary differential equations to describe the dynamics of the process. This is what computer people can understand, and this is what's used to be modeled. Of course, these models can get quite complicated. I just summarized here some of the things that we need to bring together in this type of modeling. So these bacteria also grow and reproduce. So we have to use these models that capture the nutrient factors from the environment. So if there is enough food, they grow and they divide. If there is not enough factor, they shrink and eventually they die. So this is captured in these factors like this factor M, and there is also this utility that describes the metabolic cost of this process, of the growing process. So all these things are clearly modeled at the cellular level. Again, everything is about the node level model in this network that I was mentioning. And as you see in this representation, we have here an equation for the EPS factor, the GUI thing around the bacteria that Luisa mentioned in her talk. So we have, again, an equation describing the dynamics of this process. And the whole secret here is to see how we can model these things and how we can use the data from the lab in order to characterize all these parameters that you see, all these constants, all these utility factors, in order to balance these kind of equations to describe the proper behavior that we want. So this is exactly the point where everything that has been going on in this conference can come together to help model this and improve these models in order to have the most accurate behavior captured that correlates well with the data observed in the lab. So once we have this network, we need to go to build the network in the cell. So once we have the intracellular network built, we need to go to the intercellular model. And this is not like modeling networks of sensors on internet when we plant those sensors and we know how they communicate and how they reach each other, because in this case, the network has to be formed. These things happen in 3D space where this kind of bacteria swim. So they have a certain range, as you see in this representation, at which they can be heard. And essentially, the range here is based on the level of the LAS-R AI factor that they have the C factor in the previous equation, which can go from low concentration to high concentration. The higher the concentration, the wider the range at which a particular bacteria can be heard. So in this representation, this red bacteria can be heard all the way around. So this bacteria and this bacteria, blue, yellow, I'm sorry, and green, can hear the message sent by this. So it's essentially the quorum sensing molecules that are sent, and they can reach the neighborhood bacteria. But the blue one shown here, the dark blue one shown here, cannot be reached because it has a very low level of quorum sensing. So in order to build the network, we look at two logical conditions. One is the level of quorum sensing. The C concentration here should be above a certain threshold, so it should be in the yellow and red region here, and the other bacteria, the partner in communication, should be reachable within that circle. So for instance, we have a link from A to B, is a bidirectional link. Why? Because both of them express high levels of quorum sensing, and during the swimming, okay, through the liquid, they happen to be in the area that is close by, so they can reach each other. As opposed to this one, green, which has still a significant level of quorum sensing, but its range is much limited, so the A bacteria cannot be heard. So therefore, the link is unidirectional here, and this is bidirectional. And by putting all these things together, we can create this beautiful dynamic network that changes in the 3D space, and we can observe links appearing, links disappearing, so the network shrinks and expands depending on the activity of the bacteria. So summarizing the entire process, it starts with bacteria colonizing a certain or attaching to a certain surface. As you see here, they start exchanging signaling molecules and they start communicating with each other. That communication helps them build this cocoon around them, so this EPS that serves as a source of food ultimately, and therefore they can also hide around this cocoon, and therefore they can hide from antibiotics, and therefore be much harder to be killed by the antibiotics. And if you remember from Luisa's presentation, she mentioned 1,000 times more difficult under a certain setup to kill the bacteria with the same antibiotics. So in this case, I'm going to use a sort of metaphor for this molecular tweeting, because this is essentially what happens, where we have three types of bacteria represented here. The red ones are the ones that are very active. They send and receive these signals, which will be sort of like Twitter messages saying, let's build this EPS cocoon around us to protect us from future attacks. And to this, some of them, like the ones shown in blue here, react differently. They are signal blind, so there are some mutants. They don't receive this signal. Imagine me putting my hands on the ear so I cannot hear anything you say. But at the same time, they send the signal further in order to create this synchronized behavior. The yellow ones are a different type of mutants that do not they receive this signal from the red and blue bacteria, but they don't send it further. So these are signal negative. So in a population, usually we have a mix of this type of contributors. The red ones are the regular ones, are the wild type that are very active and have a sort of very altruistic behavior. The other ones are cheaters, and therefore the kind of networking that happens becomes skewed one way or another, and some of them will be more favored by this EPS, source of EPS, others will be less favored, so therefore they may eventually be overcome by the other cheaters. So in the simulation environment that we have the ability to simulate multiple layers, as you see here, and two important phases. One is a biological phase, when these agents grow, okay, and multiply. And the physical phase here is the one when they arrange themselves in a 3D space such that not two of them will occupy the exact same position in the three-dimensional space. So a little bit of detail from the computational side. As a function of the number of bacteria, as you see here, the number of iterations required to stabilize, to find the steady state behavior of this kind of arrangements increases significantly. So the physical phase has a significant contribution to the computational aspects of the problem. And this shoving algorithm that is used to do this can be actually fine-tuned depending on the level of precision that we allow in these overlapping calculations in order to speed up or to slow down the computations and therefore make them more accurate. In reality, this kind of simulations, in order to have any biological significance, have to be significantly speed up by the hardware. In this case, we show some examples that we built based on the GPU platforms, which allowed us a significant speed up of the shoving algorithm for the physical location calculation. Typically 2 orders of magnitude improvement and speed up. And this way we are able to simulate something that will be in the range of 15, 20, 30 minutes. That will be an equivalent of 1 day of biofilm growth in the lab and observe the results in our simulator. This is basically based on exploiting the parallelism that is inherently available in this type of calculations. And probably there is a lot more potential in order to parallelize this and obtain even more in these settings. This is an example of the biofilm growth which involves only the wild type shown in red. You see here how small of a space, 3D space is this. So from a biological standpoint, this is almost irrelevant, but it's relevant to show the concept, how we model. And you see the EPS here and there that helps this growth, supports all these wild type cells. They all participate in this communication. They all receive messages to help build this EPS, and they all act accordingly. So as you see, after a number of hours, these 3D structures, the biofilm that Luisa showed in real life, can be observed in simulation. And the interesting thing is here that once we have this kind of representation, we can do anything we want in order to observe it from different angles, under different physical-chemical conditions, so on and so forth. So one particular example is shown here where we have a mixed population. In other words, we have the red ones, the wild type, but also the blue and the yellow ones, as shown here with these colors. And you see that the communication leads to a lot of dynamic changes in the overall structure of this biofilm. First of all, we're talking about 100 to 200 microns depth in this 3D mountainous structure. And you see how detailed type of analysis can we do once we build the structure and to see how these molecules are arranged one on top of another, so on and so forth. The interesting thing is you see that the blue ones are pretty much suffocated by the red ones, and this is because the EPS that grows, although it's a public good, because of the physical location, it cannot help the mutants that are remotely. So this is how bacteria actually avoid the tragedy of the commons, and the ones that dominate are the red ones. More interestingly, in this type of structure, I want to point out that if you look carefully in this representation, which is exact same scenario, but in this case we highlight the links with this different color here, you'll see that most of the links in the network appear actually in the regions that are dominated by the red bacteria. In other words, there is where the most intense communication happens in order to build this biofilm. So this is a completely different perspective to what can be observed by pure experimental biological in a biological setting, because it shows this kind of networked life that happens at nanoscale, and all these communities have a very different type of behavior under the same type of conditions in terms of food that is provided by the environment. So you see these links, okay, that appear under this, between these molecules, and these are, once we have these links and once we extract them, we can take them offline, which we actually did, and we analyze them with GFI tool. For those of you familiar, that is used pretty much everywhere in social networks analysis. So the way in which I and you communicate on Twitter is analyzed by this GFI, and we look at the communities, at the size of the communities, and this kind of interactions. We did the exact same thing as we shown here, and these are some of the data that we could observe by using this under GFI analysis. I'm going to highlight some of the results. So we have data about the physical appearance of the biofilm. We see the biofilm growth, the thickness, the roughness, but also a lot of metrics that are relevant from a network perspective. In other words, you see here the growth of communities, the links here, the clustering, and very much to our surprise, we realized that bacteria are organized in communities that are on medium size rather than large size, so they don't mix in a large ginormous component simply because there is too much diversity in the genetic material, in their physical conditions, and the way in which they express these molecules and communicate with each other. And you see that the interesting moments, the critical moments when the number of links that is generated saturates here coincides with the, ultimately with the virulence, okay, that is expressed here, and with the network, this is the clustering coefficient that shows that the network is stable and it's formed on some well-defined communities, as I said, of medium size. Interestingly enough, we try to play a little bit more on the funny side of the story. We try to see what other networks from real life will look similar to this network of bacteria. And you see here a variety of networks, technological and social networks. And at the end of the day, the one that was the closest in terms of pure network metrics perspective was the one that was based on James Bond franchise, where two actors are connected together if they played at least in one James Bond movies in the last 50 years of existence. So another network that we found was the World Transportation Airlines, which had actually even closer data in terms of network clustering parameters, community size, so on and so forth. So by no means this shows that bacteria are intelligent, but it's interesting because I imagine in this franchise, the distribution of factors played a lot of politics, and a lot of relationships changed the actual distribution in the last minute. So it's interesting to see that the similarity is much higher there compared to, say, internet or power grid or all these networks that are frequently analyzed these days. So once we build this network, we try to look at what Luisa was mentioning earlier to see how we can dismantle this network. So the quorum sensing inhibition. So in other words, if we add these quorum sensing molecules on them, what will change? In other words, if we don't go by the standard antibiotics, which the mission is to kill all the nodes in the bacteria, how about if we go selectively about leaving the nodes as they are, but dismantling the connections among them? So instead of wiping out the entire population of bacteria, good and bad bacteria, how about if we focus just on the links and therefore target specific ways to interrupt this network? And these are three ways in which we tried in these simulations. So this one is to try to alter these molecules here. So this will be to change the molecules such that they are harmless when they attach to form this LASAR complex. Another way was to interrupt the communication of the receptor side, and finally to attack it at the source, at the gene source, which of course is the most efficient. The interesting thing is if you see the virulence, how it decreases once the quorum sensing inhibitor is attached, it's coming back after a while, and this is exactly the antibiotic resistance phenomena that you see. So after a while, bacteria, after multiple applications of these quorum sensing inhibitors at different points in time, bacteria learn how to defend and learn how to protect themselves, and they will come back sooner or later even stronger. So from this perspective, we thought, well, probably finding a new antibiotic to cure infections will only offer us a delay in the process of having to face them again, because sooner or later they will learn, and usually this process happens very soon. It's typically months after the antibiotic is created. They learn how to defend themselves. And I'm not going to go into these details, but once we model this process, it's very interesting because it can be formulated as a game theory problem, and you can see various ways in which these populations of bacteria react to this kind of inhibitors. And this is the image that I would like you to see. As Luisa said, this is when the quorum sensing disappeared completely. This is when it was very active, the green one, and this is how the data looks on the simulation. So it's sort of side-by-side comparison. What can you see in the lab and validate the data, and what you can do in a simulation environment when you have a lot more flexibility and you can change the parameters on the fly and see how that influences the overall behavior. In the last 5-6 minutes, I want to show just to hint at some possible applications of this. So once we build this network that connects all these cells at nanoscale level, what can we do about it? Well, one idea would be, for instance, to try to control the network. In other words, instead of applying chemicals that are one-shot type of application to dismantle the network, how about if we introduce some biological controllers that can act only when needed and as long as it's needed? In other words, how about if we engineer some cells? That means either use purely synthetic cells or just engineer regular bacteria, say like E. coli, and make it act as a switch that activates these quorum sensing inhibition molecules when needed to dismantle the network, and when the network is not a problem because, for instance, the density of the quorum sensing molecules is low enough, just to shut down. It's similar to having sensors in this room say that when people get in, the lights turn on, and when the people get out and nobody's moving anymore around this space, the lights go off automatically. So this will be a sort of metaphor. And we did such a controller. We synthesized such a controller. You see the equation that kind of summarizes this idea. The setup is pretty much the classical control where you have a negative feedback that you explore and you try to keep the level of the to control the level of the quorum sensing molecules based on a certain threshold that you feel is okay and is tolerable. And of course, in this case, you see all the translation of the electrical parameters from classical control theory into this environment. And just to give you a sense of complexity, we synthesized this controller using completely available parts from the parts registry. And the complexity is about equivalent 2 times the complexity of 1-bit full adder that you will synthesize, or the complexity of 4 inverters that will be synthesized, you'll be synthesizing using these biological parts. So the overhead is very small, about 4,000 pairs, but it's very interesting that the behavior is actually stable. And you see the 2 stable states, that monostable high and monostable low, that can be controlled very precisely when this controller is applied, and is a very sharp decrease that remains pretty stable over time. More interestingly, you see after when the controller is put here, there is no comeback of this, so as opposed to this when you just use the classical inhibition. So the controller can quite stably fix the level of molecules below a certain threshold, of quorum sensing molecules. More interestingly, for those of you into control issues, normally you can observe a quasi-stable state that is shown here in green, and when you enter this controller, this state disappears completely, and if you look at the x-axis, it becomes 10 times more stable in terms, is like the threshold is increased significantly, so you can have a very sharp transition between high and low modes that will activate this switch. So it's a possible interesting application. And finally, as I mentioned in the very beginning, this offered us a piece of different flavor for thinking about the architectures that we can use to do this kind of simulations. As of now, we use off-the-shelf boards from NVIDIA, but when we realized that this type of calculations are not precise, should not be done with extreme precision, we realized that this in fact can stimulate approximate computing, platforms that do these computations approximately, not necessarily from a value point of view, which was the classical approximation theory, but also at the structure level. In other words, you can work with trees, say, that have some branches missing because their contribution to the overall picture is not critical. So what we did here in this plot, and we show in these plots, we try to see on a multi-core platform with multiple threads that we can allow for different algorithms. This is the k-means and this is the breadth first search. How much potential we have for optimization compared to absolute Amdahl's law here, and you see the potential increases significantly. Is a huge gap that is completely unexplored. And the interesting thing is, if we go into this problem space, it could lead to a completely new metaphor for the future multi-core platforms to do this kind of simulations. So coming back to the example that I showed earlier, when you combine this CPU with GPU, not only that we can eliminate completely these global transfers through the, from the memory to the computing units, we can keep everything localized if we keep the application at hand and do this global transfer only once, but everything after that remains localized, so it leads to a significant speed up and efficiency in computation. So this is a wide open area that can be explored, and there are many reasons to explore this, not just the infection side of the story that we cover in this part of the special session, but also if you go in other areas, like for instance cancer cell modeling that you'll see in the second part, also microbiome, because this kind of computational microscope that we can build and use to allow biologists to see things that are hard to see in the lab, or sometimes impossible to see in the lab, can lead to an exchange of ideas in a completely different plane that was never possible before. So in my presentation, I just show how neatly this data can be displayed, how fast can be this done compared to the real-time lab simulation. In other words, we can do many simulations, lab experimentation, I meant. So we can do many simulations, and only the most interesting ones actually be taken to the lab for representation, and we can do things infinitely complicated once these models are solid and grounded in the biological reality. So in conclusion, I think this problem space is extremely promising and should be one of the main concerns for computer scientists and computer engineers. And focusing on the communication between the cells rather than communication inside the cells is the future, because this can lead to interactions at the tissue level, at the organ level, at the system level, and probably if we project where the biology is going to be 20 years from now, it's going to be more like a science of systems than a science of pure experimentation. Thank you very much.